In case of module 3, respiration in plants, the respiratory balance sheet of ATP. So, if you recollect this, the information with regard to the glycolysis and Krebs cycle. So, total oxidations, which are the 6 in glycolysis 1, in between glycolysis and Krebs cycle 1, in Krebs cycle it is the 4. In all these total oxidation, 6 oxidation, 12 coenzymes are involved. In these 12 coenzymes, 10 are NADS2, 2 are FADS2. Even in the 10 NADS2, 2 formed in glycolysis and 8 in mitochondria. And 8 in mitochondria, 2 that refers to in between glycolysis and Krebs cycle. And 6 for a 2 turn of the Krebs cycle. So, in calculating this number of ATP, we can make calculations how many net gain of ATP for one glucose molecule oxidized. But in reality, it remained a theoretical exercise. Not in case of the practicality, it is not valid. But anyhow, there is a beauty in extracting this energy by the living system and trapped in the form of ATP. So, in reality, it is a theoretical exercise. The theoretical exercise is based on certain assumptions. The first assumption is the process or reactions occur one after other. That is, one substrate converted into other substrate, the glycolysis, Krebs cycle, electron transport system, or the glycolysis, link reaction, Krebs cycle, electron transport system. That is one after other, the first assumption. The second assumption is glucose is only the respiratory substrate, no other substrates are participating at any stage. That is the second assumption. And third, whatever the NADS2 formed in glycolysis, for oxidation, it enters into the mitochondria. Next, the fourth assumption, no substrate is withdrawn when the glycolysis or link reaction or the Krebs cycle, no substrate is withdrawn. These are all assumptions. Why these assumptions are not valid in a living system? It is not valid, it is not the truth. Because the process do not occur one after other. It is simultaneous, not glycolysis, link reaction, Krebs cycle, electron transport system. The process, it is simultaneous. Second, glucose is not only the substrate. Of course, the glucose participated first step. But some other substrates are also participating, but not at first step, but some other steps. If you take the fats and protein, pure fats, pure protein never be substrate. But after certain chemical modification, the protein and fats entering in respiratory pathway at some other steps. So, which are also respiratory substrate. Next, the substrates are withdrawn as and when required to synthesize other biomolecule. Suppose in case of the Krebs cycle, alpha ketoglutarate acid is converted into succinyl coenzyme A. If it is a continuous succinyl coenzyme A, succinic acid, fumaric acid, malic acid, oxaloacetic acid, and acetyl coenzyme A is formed. But whenever it is required, the alpha ketoglutarate acid is withdrawn, and by reductive amination, it is going to form amino acid. It is required by the cell. So, substrates are withdrawn as and when required. And further, these reactions are enzymatically controlled, and enzymatic reactions are controlled by multiple means, temperature, pH, and other things are present. So, anyhow, there is a beauty in extracting the energy by the living system and trapped in chemical bonds, that is ATP. And finally, for one glucose, the net gain of ATP is going to be 38 ATP. So, before doing such calculations, so in case of the Krebs cycle, there is no involvement of oxygen, direct involvement of oxygen. In case of the glycols, there is no involvement of oxygen. But even then, oxygen role it is vital. It drives the whole system by removal of hydrogen, and it is the final acceptor of the hydrogen. Unless it accepts, it is not supposed to remove the hydrogen from the system. So, oxygen has a terminal role. What is the terminal role? It is a vital. It drives the whole system by removal of hydrogen from the system, and finally it acts as a hydrogen acceptor. So ultimate electron acceptor, ultimate hydrogen acceptor is oxygen in aerobic system. Next. So in case of NADS2 and FADS2 in electron transport system, for one NADS2 it is going to yield 3 ATP. For one FADS2 it is going to yield 2 ATP. So how many coenzymes are involved? 12 coenzymes are involved. Suppose one NADS2 in oxidation in aerobic respiration in electron transport system, its pathway is complex 1, UQ, complex 3, cytochrome C, complex 4, oxygen with formation of water molecule. For one NADS2, one oxygen atom is required or photo is required with formation of one water molecule by the ejection of 4 plus 2 10 proton into intramembrane space. You have to recollect this information. So 1 NADS2 3 ATP, 1 FADS2 2 ATP. And you go for simple to complex with regard to the calculating the number of ATP. Suppose if you take the glycolysis, for one glucose molecule, how many number of ATP? 4 ATP. How many ATP are utilized in first and third reaction? 2 ATP. So net gain of ATP is going to be, it is the 2 ATP. Next. If glucose 6-phosphate is the respiratory substrate, not glucose, as glucose is the substrate, 1 ATP is utilized. If glucose 6-phosphate is the respiratory substrate, it is hypothetical. How many net gain of ATP? It is going to yield 3 net gain of ATP because first 1 ATP is not utilized. If fructose 6-phosphate is the respiratory substrate, net gain of ATP is going to be, it is also going to be 3 ATP because glucose 6-phosphate converted into fructose 6-phosphate is isomeric change. If fructose 1,6 biphosphate directly participate in this, a total of the 4 ATP is formed. And we are not going to call any net gain of ATP because there is no consumption. So, total number of ATP formed from one molecule of fructose 1,6 biphosphate 4. Because one molecule of fructose 1,6 biphosphate is going to yield 2 G3P molecule. Per 1 G3P molecule, 2 ATP are formed. Where the 2 ATP are found, 1, 3 biphosphoglyceric acid to 3 phosphoglyceric acid, 1 ATP. And finally, phosphoenol pyruvic acid to pyruvic acid, 1 ATP. So, per 2 G3P, how many ATP are formed? 4 ATP are formed. So, total ATP, 4 ATP, net gain of ATP, it is the 2 ATP. Suppose the question it is like this. How many number of ATP, that is net gain of ATP formed in glycolysis with involvement of oxygen? There is no involvement of oxygen really in case of the glycolysis. As earlier said, the NADS2 enter into the mitochondrial matrix is going to oxidize to yield what it is ATP. As the process do not occur one after other, it is simultaneous. The question it is, how many net gain of ATP formed in glycolysis with involvement of oxygen? As in glycolysis, net gain of ATP is the 2 ATP. In these 2 NADS2 are formed. This 2 NADS2 on oxidation in mitochondria is going to yield how many ATP? 6 ATP. So net gain of ATP in glycolysis per one glucose molecule with involvement of oxygen is 8 ATP. Next, how many total number of ATP in glycolysis per one glucose molecule with involvement of oxygen? As total ATP in glycolysis is the 4. Oxygen is involved only to oxidize what NADS2 formed in case of the glycolysis. So, as it is going to yield what? 6 ATP plus 4. Total it is the 10 ATP. Next. In case of the link reaction pyruvic acid to acetyl coenzyme A. So for 2 pyruvic acid, how many NADS2? The 2 NADS2. 2 cycles, TCA cycles. How many NADS2? 6 NADS2. How many FADS2? 2 FADS2. Next. As total 10 NADS2 are present, formed 2 FADS2. As earlier said, 1 NADS2 is going to give 3 ATP. So 10 NADS2 is going to give 10 into 3, 30 ATP. Next. 2 FADS2 formed in case of the Krebs cycle. 1 FADS2 is going to give 2 ATP. 2 FADS2 is going to give 4 ATP. So 34 ATP formed due to oxidation of only coenzymes per 1 glucose molecule. That is one thing. 34. So net ATP formed in case of the glycolysis is the 4. 34 plus 4, 38. In case of the 2 terms of the Krebs cycle, 2 ATP. 38 plus 2, 40 ATP. So total number of ATP formed in aerobic respiration per oxidation of 1 glucose molecule is 40 ATP. So net gain of ATP in aerobic respiration per 1 glucose molecule, as earlier 1 and 3 reaction in case of the glycolysis, 2 ATP are consumed. So net gain of ATP is going to be 38 ATP. So total ATP 40, net gain of ATP is going to be 38 ATP. Next. Suppose if you take the pyruvic acid, acetyl coenzyme A, 1 NADS2. Okay. And in case of the TCA, the Krebs cycle, 1, 2, 3 point. At 3 point, at each point 1 NADS2. So there are 3 NADS2, 1 FADS2, 1 ATP. So for 1 pyruvic acid, suppose if you take the mitochondria, how many NADS2? That is the 4 NADS2, 1 FADS2, 1 ATP formed in case of the Krebs cycle. Suppose if you want to count this number of ATP due to oxidation of coenzymes, you should not consider the ATP obtained in substrate level phosphorylation. Suppose if you want to calculate the number of ATP due to oxidation of the substrate, whether glucose or some other substrate, we have to consider the ATP formed in substrate level phosphorylation also. Next. So, for 1 pyruvic acid, the 2 type of the question, oxidation of 1 pyruvic acid or oxidative breakdown of 1 pyruvic acid in mitochondria. How many number of ATP are formed? For 1 pyruvic acid, the 4 NAD. So 4 NAD means 4 into 3. That is the 12 ATP. 1 FAD, that is going to 2 ATP. And 1 ATP formed in case of TCA cycle at succinyl coenzyme to succinic acid, SLP, 1. So 15. So oxidative breakdown or oxidation of 1 pyruvic acid in mitochondria, it is going to be 15. Next. So oxidation of the 2 pyruvic acid molecules, it is going to be 15 plus 15, 30. And if you consider these 2 NADS2 of glycolysis, 6 ATP, 36. And in 2 ATP in glycolysis as a net gain, 6 plus 2, 8. So 30 plus 8, 38. So net gain of ATP is going to be 38. Next. Oxidation of 1 pyruvic acid in Krebs cycle is going to yield how many number of ATP? As you are talking, oxidation of pyruvic acid not in mitochondria in case of Krebs cycle. 3 NADS2, 9 ATP, 1 FADS2, 2 ATP, and 1 ATP in substrate level, it is going to be, it is the 12. So oxidation or oxidative breakdown of 1 pyruvic acid in Krebs cycle is going to be 12. The same question, just modification. How many number of ATP formed due to oxidation of coenzymes per 1 pyruvic acid in Krebs cycle? You should not consider the SLP. So 3 NAD, 9, 1 FAD, 2, is going to be 11 ATP. So in these two questions, oxidative breakdown of one acetyl coenzyme A or one pyruvic acid in Krebs cycle is going to be 12. Oxidative breakdown of one pyruvic acid means it is the 12. Okay. Oxidation of coenzymes for one pyruvic acid means it is going to be 11 in case of the Krebs cycle. Okay. Next. How many number of ATP formed in mitochondria alone per one glucose molecule in aerobic respiration? As earlier said, in this mitochondria, 15 per one pyruvic acid. Another pyruvic acid is going to be 15. It is 30 formed in mitochondria. As NADS2 formed in case of the glycolysis, in order to oxidize to yield ATP, it must enter into the mitochondria. It is going to yield 6 ATP. So number of ATP formed in mitochondria alone is going to be 36 ATP. Like this. Next. Oxidative breakdown of one phosphoenol pyruvic acid is going to yield how many number of ATP? So PEP is going to give pyruvic acid. In that step, we are going to get 1 ATP. In this earlier said in mitochondria, for one pyruvic acid 15. And we have to consider for one PEP, that is another ATP, 16 ATP. And even for 2-phosphoglyceric acid, even for 3-phosphoglyceric acid, we are going to consider 16. Suppose 1,3 biphosphoglyceric acid, one molecule of 1,3 biphosphoglyceric acid, oxidation going to give how many number of ATP? In mitochondria 15, that is, okay, 1,3 biphosphoglyceric acid to 3-phosphoglyceric acid, 1 ATP. Phosphoenol pyruvic acid to pyruvic acid, 1 ATP. 15 plus 2, 17 ATP. If it is 1,3 biphosphoglyceric acid per one molecule. Next, per 1 G3P, how many number of ATP formed due to oxidative breakdown of 1 G3P molecule in aerobic respiration? As in case of the mitochondria 15, it is constant. Per 1 G3P, how many ATP are formed? 2 ATP are formed. For 1 G3P, how many NADS2 formed? 1 NADS2. We have to consider the 15 plus 2 ATP in glycolysis, that is 17. And 1 NADS2 is going to give 3 ATP, 20. So, oxidation of 1 G3P or oxidative breakdown of 1 G3P is going to yield how many number of ATP? 20 ATP. For another G3P, 20. So, total it is going to be 40 ATP. So, 1 G3P question. Suppose just modify this question 1 G3P. How many number of ATP formed due to oxidation of coenzymes? The question it is oxidation of only coenzymes for 1 G3P in aerobic respiration. We should not consider the SLPs. So earlier we said for 1 G3P total it is the 20 ATP. And in 2 ATP in case of the glycolysis substrate level and 1 ATP in case of the Krebs cycle, you should not consider. So, how many number of ATP formed due to oxidation of coenzymes for 1 G3P in aerobic respiration? 17. How many number of ATP formed due to oxidative breakdown or oxidation of 1 G3P in aerobic respiration? 20. So, 20 per one question, 17 per what it is the other question. Like this, we can calculate this number of it is ATP. Suppose, how many number of ATP formed for oxidation of all reduced coenzymes? It is going to be 34. All NAD coenzymes, it is going to be 30. All FAD coenzymes for one glucose, it is the 34. And with regard to the oxygen atoms in molecule, how can you calculate it? For total oxidations, it is the 6. Number of coenzymes, it is the 12. For each NADS2 or FADS2, one atom of oxygen it is required. As the 12 coenzymes are involved, 12 coenzymes is going to oxidize. The NADS2 is oxidized by oxygen. So 12 reduced coenzyme requires 12 oxygen atom. That means 6 oxygen molecules. Suppose for one pyruvic acid, how many oxygen atoms are required? For one pyruvic acid in mitochondria, one pyruvic acid acetyl coenzyme, one NADS2. In this circle, 3 NADH2, 1 FADH2. Total at a 5 reduced coenzyme. So 5 atom of oxygen, 5 of O2s are required. Suppose for 2 pyruvic acid, how many oxygen molecules are required for oxidation of pyruvic acid in aerobic respiration? For 2 pyruvic acid, the 5 reduced coenzymes and another 5 for another pyruvic acid. Total it is going to be 10 reduced coenzyme. For 10 reduced coenzymes, how many oxygen atom? 10. How many oxygen molecule? 5. So 10 oxygen atom, 5 oxygen molecule. So for 12 coenzymes, how many oxygen atom? 12. How many oxygen molecule? 6. How many metabolic water found for all oxidation of the coenzyme? 12 water molecules are found. So finally, NADH2 is going to be oxidized by oxygen. Final electron acceptor, it is oxygen. Next, in next paragraph information with regard to fermentation and aerobic respiration. In fermentation, partial oxidation of the glucose with a few number of ATP, that is a 2 ATP as the net gain in case of fermentation. As in case of aerobic respiration, complete oxidation of the glucose with the release of the carbon dioxide and formation of this water with a large number of ATP. That is, earlier we said 38 net gain of ATP, total ATP is the 40 ATP. In this last point, the oxidation of NADH2 rather slow in alcoholic fermentation as well as lactic acid fermentation. But oxidation of NADH2 is so vigorous in case of aerobic system. You have to recollect it. In alcoholic fermentation, NADH2 oxidized by, it is acetaldehyde. In lactic acid fermentation, NADH2 oxidized by, it is the pyruvic acid. In aerobic respiration, NADH2 oxidized by, it is oxygen. So in 3 cases, alcoholic fermentation, lactic acid fermentation, and in case of aerobic respiration, NADH2 is going to be oxidized. But thing is, in lactic acid fermentation and alcoholic fermentation, NADH2 is oxidized and not going to yield any ATP in case of alcoholic fermentation and lactic acid fermentation. But in case of aerobic respiration, NADH2 undergoes oxidation, is going to yield ATP, that is called oxidative phosphorylation. So we covered the aspects related to the balance sheet of ATP in case of respiration process. In next module, we are going to cover the amphibolic pathway, and it is RQ, and some related questions with regard to this the total respiration. So subscribe and share.